before we move on to the next forensic application, let's first understand what chromatography is. Chromatography is a laboratory technique used to separate the components of a mixture based on how fast each component moves through a system. It is widely used to separate, identify, and analyze different chemical substances, especially when dealing with complex mixture. Chromatography works using two phases. The first is the stationary phase, which remains fixed in place. The second is the mobile phase, which moves through the stationary phase carrying the sample. Since different compounds interact differently with these two phases, they travel at different speeds and become separated. In forensic science, investigators rarely receive pure samples from a crime scene. Instead, evidence such as blood, urine, drug samples, or fire debris is often contaminated or mixed with many other substances. Chromatography allows forensic scientists to separate this mixture, identify the substance present, and provide reliable scientific evidence that can be used during the criminal investigation and in court.
Next is principle of chromatography. There are two principles of chromatography, which are stationary phase and mobile phase. Stationary phase remain fixed inside the chromatography column or plate. As for mobile phase, it moves through the stationary phase carrying the sample. So each compound has unique chemical properties, so some compounds are more strongly attracted to the stationary phase and move more slowly, while others are more attracted to the mobile phase and move faster. As a result, the compounds separate from each other, allowing scientists to identify each substance individually. Let's go through how does chromatography work. The process works as follows, such as the sample is introduced into the chromatography system. Next one is the mobile phase carry the sample through the stationary phase. Each compound interact differently with the stationary and mobile phase, such as compound with strongest attraction to the stationary phase move more slowly, while compounds with a strongest attraction to the mobile phase move more quickly. As the compound travel at different speeds, they separate into individual components. A detector records the separated compound, producing a chromatogram. Scientists compare the chromatogram with non-standard to identify the drug or toxic substances present. In forensic drug analysis, two of the most commonly used chromatography techniques are gas chromatography (GC) and high-performance liquid chromatography (HPLC). Gas chromatography uses a gas as the mobile phase and is ideal for analyzing volatile compounds such as certain drugs and poison. It is often combined with mass spectrometry, forming GCMS, which provide highly accurate identification of chemical substances. On the other hand, high-performance liquid chromatography (HPLC) uses a liquid as the mobile phase. It is suitable for analyzing non-volatile or heat-sensitive compounds and is commonly used to detect drugs in biological samples such as blood, urine, and tissues. So, why is it important? This is because it can identify illegal drugs or toxic chemical, determine the type and amount of drugs present, analyze evidence from blood, urine, tissue, beverages, or powders, and it provides reliable scientific evidence that support criminal investigation and court proceedings.
Next, I'll explain the methodology used in forensic drug analysis. First is sample collection. This is a very important step because the accuracy for the analysis depends on collecting samples properly to avoid contamination, degradation, or tampering. These are two types of samples. The first is trace material, such as street drugs in the form of powders, tablets, or liquids. These are collected in anti-static tempered evidence bags to preserve the evidence. The second is biological specimen. For example, blood is collected in the tubes containing sodium fluoride and ETA to prevent clotting and preserve the sample, while urine is collected in sterile containers. Next is sample preparation. Solid samples are first homogenized to make them uniform. Then, solid phase or liquid phase extraction is used to isolate the drug compound, and the sample's pH is adjusted using specific solvent. If the analysis uses gas chromatography, derivatization may be performed to make certain drugs more stable and suitable for analysis. Finally, we have a detection using mass spectrometry. First, the molecules are ionized by electrons or chemical ions. These ions are then separated according to their mass to charge ratio using an electromagnetic field. Lastly, the detector records the abundance of each ion, producing a mass spectrum that helps identify the drug accurately.
After collecting the evidence, the sample will analyzed in the forensic laboratory using chromatography techniques, specifically gas chromatography, mass spectrometry, GCMS, and high performance liquid chromatography, also known as HPLC. These techniques separate the different compounds present in the sample based on their chemical properties. The separated compounds produce a chromatogram which show the different substances detected in the sample. By comparing the chromatogram with known reference standard, forensic scientists can identify the specific drug or toxic substance present. In this case, the analysis detected a toxic drug in the victim's blood sample, confirming that poisoning was the cause of death. The result from chromatography provided important scientific evidence that helped investigators understand what happened and supported the criminal investigation. For this case study, we will look at how chromatography helps solve a suspected poisoning case. A victim was found dead under suspicious circumstances, but there were no obvious signs of physical injury. Due to the unusual situation, investigators suspected that the victim may have been poisoned. During the investigation, forensic scientists collected important evidence, including blood and urine samples from the victim. They also collected possible sources of the poison, such as a drink of medication found at the crime scene. These samples were then sent to the laboratory for further analysis using chromatography techniques.
Beverage collected at a crime scene can provide important forensic evidence because they may contain alcohol, medications, or illegal drugs. Investigators collect and seal the drinks carefully to preserve its integrity. In the laboratory, alcohol is commonly analyzed using head-space gas chromatography (HGCFID), while suspected drug or medications are identified using DCMS or LCMS or MS. A recent real-life example is the death of actor Matthew Perry in 2023. After he was found unresponsive at his home jacuzzi, forensic toxicologists analyzed his biological sample using advanced techniques such as liquid chromatography, mass spectrometry (LCMS or MS). The analysis detected a high concentration of ketamine, which the medical examiner determined was the primary cause of his death. Following the toxicology findings, investigators launched a criminal investigation. In 2024, two doctors and several other individuals were charged by U.S. federal prosecutors. They were accused of illegally supplying Perry with a large amount of ketamine outside accepted medical practice, despite knowing the potential risks. This case demonstrates how chromatography plays a vital role in forensic science. It provides accurate and reliable evidence by identifying drugs in biological samples, helping investigators to determine the cause of the death and supporting the criminal investigator with scientific proof. To conclude, chromatography is one of the most important analytical techniques in modern forensic science, particularly in drug analysis. Crime scene evidence and illegal drugs are usually recovered as complex or contaminated mixture rather than pure substances. Chromatography makes it possible to separate this mixture and isolate the individual chemical compounds to accurate identification. This high level of accuracy helps forensic scientific produce reliable results that can support investigation and legal proceedings. More importantly, it helps ensure justice by identifying the correct evidence, protecting innocent individuals from wrongful conviction, and providing scientific proof that the court can trust. Overall, chromatography plays a vital role in forensic investigation because it provides the scientific certainty needed to solve crimes accurately and fairly.
So this is our references for our topic, which are chromatography in action that solving crimes through drug analysis.